There are many processes in living organisms that involve electric potential differences. In this final section of Chapter 19, we will explore two of these. First, let's consider the physics of action potentials. These occur in several types of animal cells, and some plant cells as well, where the potential in a cell membrane changes rapidly. The figure shows the anatomy of a typical nerve cell. A nerve consists of a bundle of axons, and each axon is one part of a nerve cell or neuron. A typical neuron consists of a cell body with numerous extensions called dendrites and a single axon. The dendrites convert stimuli, such as pressure or heat, into electrical signals that travel through the neuron. The axon sends the signal to the nerve endings, which transmit the signal across a gap, called a synapse, to the next neuron or to a muscle. A resting neuron is one that is not conducting an electrical signal. The change in the resting membrane potential is the key factor in the initiation and conduction of a signal. When a sufficiently strong stimulus is applied to a given point on the neuron, gates in the membrane open and sodium ions flood into the cell, as the figure illustrates. The sodium ions are driven into the cell by the attraction to the negative ions on the inner side of the membrane, as well as by the relatively high concentration of sodium ions on the outside of the cell. The large influx of sodium ions first neutralizes the negative ions on the interior of the membrane and then causes it to become positively charged. As a result, the membrane potential in this localized region goes from minus 70 millivolts, the resting potential, to about plus 30 millivolts in a very short time, as the figure shows. The sodium gates then close, and the cell membrane quickly returns to its normal resting potential. This change in potential from minus 70 millivolts to plus 30 and back to minus 70 millivolts is known as the action potential. The action potential lasts for a few milliseconds, and it is the electrical signal that propagates down the axon to the next neuron or to the muscle cell. The typical speed of an action potential is approximately 50 meters per second, or 110 miles per hour. As a final topic of this section, let's consider the medical diagnostic technique of electrocardiography, which takes advantage of the fact that the surface of the human body is not an equipotential surface. Between various points on the body, there are small potential differences of approximately 30 to 500 microvolts. The figure shows electrodes placed on the body to measure potential differences in electrocardiography. The potential difference between two locations changes as the heart beats and forms a repetitive pattern. The recorded pattern of the potential difference versus time is called an electrocardiogram, ECG, or EKG, and its shape depends on which pair of the points in the picture, A and B, B and C, etc., is used to locate the electrodes. The figure also shows some EKGs and indicates the regions P, Q, R, S, and T associated with specific parts of the heart's beating cycle. The differences between the EKGs of normal and abnormal heartbeats provides physicians with a valuable diagnostic tool.